Okay, so welcome to, welcome to today's session. See, in this session, we'll be focusing on retrieval of gene sequence. Now, I've written retrieval of gene sequence. You are already aware of the nucleotide databases that were told to you or that are there. The primary nucleotide databases, that is EMBL, DDBJ, and GenBank. So we'll be focusing on how to take out the sequence from GenBank, and we'll be looking at different databases also that are present in NCBI. Apart from it, will be, as we analyze the complete database of Swiss Prot, we'll be analyzing the complete database page of GenBank. We'll be knowing what are coding sequences. We'll try to understand what are your UTR regions, how many mRNA variants are being made, what are the number of exons that are there, what information can we harness from that particular page. Fine. And also we will be talking about, in the later section, a brief about the alignment. How now we have established understanding that we can retrieve the sequence from Swiss Prot for proteins. We can retrieve the sequence of nucleotide or gene from GenBank. Then what to do with those sequences? We will be going a step, we'll be taking a step towards advanced part by understanding the basis of alignment. And once we have understood the alignment, please try to understand. Once we have understood the basis of alignment, then and then only you will be able to use the tools that are present and that will be showcased to you in the coming lectures. So that is the main objective of today's lecture. Now, before we go to today's lecture, as we had discussed earlier, we will be talk a lot about protein databases where we had understood the sequences are coming M, V, L, K, S, T. Now, the question comes from your side that what are these letters? So see, before starting bioinformatics, in fact, you should be very clear regarding the single letter codes. If we are talking about the gene sequence, the DNA, it's very simple. It's ATGC. These are the four bases that we'll be talking about. In terms of DNA or in terms of RNA, we'll be talking about AUGC. It's okay. There is no doubt in that scenario. But when it comes to amino acids, we have 20 amino acids. 20 amino acids in literature, in biochemistry, are represented in words in three-letter codes like cis for cysteine, SER for serine. But when we are going for bioinformatics, it's very difficult to accommodate three alphabets. Why? Because the memory will be very high. I have to accommodate large memory. When I'm trying to match suppose 1 lakh bases, I'm trying to match 1 lakh amino acid. This matching will not be profound. So I need a single letter code, and that single letter code is utilized to build every information in bioinformatics. So just in brief, let us talk about all those 20 amino acids and their single letter. You are very much clear of the proper names of the amino acids, right? So let's begin, and I'll show you the shortcut also. This will be the best shortcut that will be coming to know, by which you will be able to iterate all the names of 20 amino acid within a snap of a finger, with a snap of a finger. So let's see. See here. We'll start with very simple form. I'm writing here, or let me first remove everything, and then let's just begin fresh with a fresh thing. So I always remember with the short forms, like we used to remember the periodic table. So that's the way I have remembered these things. It is helping me till now, from since the point we have I've learned them. So I hope that will be very useful for you too. So this is how we go about, but in a pretty simple manner. You can also use this nomenclature the way I am writing it. And the reason why I've clubbed this, I'll also show you. So this is the thing. These are the 20 amino acids. You can count them: 2, 3, 4, 5, 6, 7, 8, 9. 9 and 6, 9 and 6, 15, 15, 16, 17, and 3, 20. Now, how I pronounce? First of all, Gavli FM with Pa. Gavli FM with Pa, Cyst and Q, or you can frame any name, Day, HRK. HRK. Now G is for glycine, G is for glycine, A is for arginine, A is for arginine, V is for valine, L is for leucine, I is for isoleucine, F is for phenylalanine, F is for phenylalanine, M is for methionine, M is for methionine, W is for tryptophan, W is for tryptophan, P is for proline, P is for proline. C, C is for cysteine, Y is for tyrosine, Y is for tyrosine, S is for serine, S is for serine, T is for threonine, T is for threonine, N is for asparagine, Q is for glutamine, D is for aspartic acid, E is for glutamic acid, and H is for histidine, R is for arginine, K is for lysine. This is the nomenclature of your amino acid. This is the nomenclature of your amino acid, and this should be by heart for every Bartek or any applied biological sciences student. This should be by heart, and there should not be any if and buts regarding this particular scenario. So, the
Excuse
why
me,
I
sir.
yeah, please go ahead.
This R is stands for
This is arginine.
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 Okay, sir.
Okay, so we can directly divide this into four sections. That's why I have written them into 4 lines. Now, what are these 4 lines? HRK, histidine, arginine, and lysine. These are positively charged amino acids. D and E, these are negatively charged amino acids. Also, you are pronouncing what? D is aspartic acid. E is for glutamic acid. Glutamic acid. Now, what is this? These are acidic amino acids. Then, positively charged is always basic amino acids. Okay, fine, no problem. Then, what is this? Cyst and Q. Okay, cyst, C, cysteine. If you see the side chain, it contains a thiol group. It contains a thiol group. STY, YST, all these three amino acids will be having a hydroxyl group in their side chain. That's why in Swiss prot, when we were studying, the phosphorylation was happening on these three amino acids. Why? Because they have hydroxyl group. And phosphate will bind to the protein via phosphodiester bond. And for that, it requires a hydroxyl group. And among all the amino acids, these three are the amino acids which contain the hydroxyl groups only. So coming to your N and Q, they both contain amino group. They both contain amino. So if you look in a broad category, what is this? These 6 amino acids are polar amino acids. Why polar? Because they have polar groups. Because they have polar groups. Now, when we talk about these, guanine, glycine, alanine, valine, leucine, isoleucine, phenylalanine, methionine, tryptophan, and proline. These all are non-polar amino acids. These all are non-polar amino acids. And among them, guanine, these are aliphatic non-polar amino acid. M is also included in aliphatic non-polar amino acid. Fine. Then among them, among all of them, which are aromatic amino acid? Answer is very simple. Answer is very simple. It's Y. Tryptophan, tyrosine, and phenylalanine. These are aromatic amino acids, and these are the amino acids which are responsible for giving you the absorbance in spectrophotometer, because the aromaticity is required for absorbance. Fine. And which is the amino acid which is required for protein folding, which actually forms a disulfide bond? For forming a disulfide bond, you need sulfur group. And sulfur containing amino acids, there are 2 amino acids which contain sulfur. cysteine and methionine. Cysteine and methionine. Among these two, among these two amino acids, only and only cysteine is the amino acid which will lead to the formation of disulfide bond, simply because it has a free thiol group. But when you see methioниne's side chain structure, you will see that after four carbons, it contains a sulfur and then another carbon. So the valency of sulfur is completely satisfied. It cannot form any further bond. That is the reason that only thiol group which is present on the cysteine is responsible for forming the disulfide bond. Okay, so this is in short a summary of amino acid, and this you should always remember while going for any bioinformatics tool, because in bioinformatics there will be no discussion regarding what is G, what is A, what is B. We bioinformatics tools expect that you are already aware what are these amino acids. So this is the in nutshell the complete detail about amino acids. So with this, let's go directly to the part where we wanted to discuss, that is the databases. Now you all already know that yes, when we are talking about the DNA databases or the nucleotide databases, you are pretty much established the understanding that there are three components that I am talking about. I am talking about the GenBank, I am talking about the DDBJ, and I am talking about the EMPL. Now, in previous lectures, in previous understanding, you already had established that seen the platforms. You have completely seen the platforms. So today we will not be talking about these platforms. We'll be using only GenBank to retrieve our sequence, and we'll be looking this in pretty detail. Now, this is maintained at NCBI, and you had a very deep, in-depth look at NCBI also. What are the different tools, different databases that are present in NCBI, and we'll have a glimpse as per our need or requirement of this particular chapter. Now, what I want to tell you in this particular section, a term I want to tell you that is called as SRS, or the technical term that is there, that is called as neighboring concept. Neighboring concept. This is in Europe, Germany. This is in Japan. This is in Japan. We are talking about. This is what we are talking about at States, right? So I'm here in India and I want to access the information. I'm working on GenBank page. I'm looking at GenBank page. I want to see what's the information about that particular DNA that is present in EMBL. So what I have to do? In normal circumstances, I have to again open a tab, open the website of EMBL, search the gene again, and look for the information. But that is again troublesome, right? Because you are maintaining the same information in GenBank and EMBL. So what the scientific community did, they made a international collaboration or consortium where they brought these three on a single platform. They brought these three on a single platform. So if I'm looking on GenBank, I'm pretty much sure that the complete information that was deposited in these two is also shared on that page, and it is visible to me, so that I need not to go anywhere else to harness the information. But if I desire also to go, I'll be having a link here which will be showcasing DDBJ or EMBL. I can click on link and I can directly enter inside the page of the EMBL. And this sharing concept is called as neighboring concept, and this retrieval system is called as sequence retrieval system. Is called as sequence retrieval system. So let us not waste much of the time. Let us directly go to the principal page that we want to study, and let's directly talk about your uh, uh, GenBank database. Let's directly go to the GenBank database. Yeah. So to open the GenBank, I'm not doing any extra thing. I'm just writing GenBank. I wrote GenBank. Please remember, it's not GeneBank, it's GenBank. It's GenBank. The first thing what it gives is an overview. Let us open the overview. Let us open the overview. And it also showcases you how to submit it. directly gives you the access of how to submit. So we will keep this how to submit to a later part. Let us directly go to Gen. So this is, first of all, this is the NCBI page. In that NCBI page, we are looking at the overview of the GenBank. Fine. So if you want to see the NCBI, you can directly go to the NCBI also, and then from the sources, you can enter inside the GenBank. So we will talk about both, how you can harness both the things. Fine. So let us talk about this. Is your nucleotide NCBI, National Center for Biotechnology Information, situated at Bethesda, Maryland, in NIH. That's why the web address came. NCBI in National Library of Medicine, National Institute of Health, government agency. So about the NCBI, you can read anything. Let us talk about the options that they have given you. They have given you the complete databases. You can go search in all the databases, and if you want to go for specialized part, you can put any specialized database in use. If I'm looking for a gene, I'll select gene. If I am looking for complete nucleotide, I'll be selecting nucleotide. If I am looking for literature, I'm just interested in reading, I'll be picking PubMed. You can pick PubMed here. If I'm looking for any chemical compound that is listed, I can go for PubChem. If I am looking for, suppose, any Mendelian inheritance pattern, I will be going for, if I'm interested in genetic study, I'll be going for OMIM. If I am interested in any kind of variation, clinical variation or single nucleotide polymorphism, I can use those things. If I'm interested in, let's say, domains which are actually conserved throughout evolution, I can use yeah, conserved domains here. I can use conserved domains. If I want to see what are the biological projects, what was the human project, human genome project, what was the mouse genome project which was done, then also I can select the specific database and can go there. So you can explore these. Exploring these options will not pamper the website, will not pamper the information there, and nobody is going to come running after you. You can go in your free time and you can surf on that. Anything you pick will be having its own description, and it's in the simplest language you can read about it. Fine. So let's start with our gene part. In the gene, let's talk about P53 that we are using. I typed P53 and I went for search. So a page will open and that page will give you the complete information. That page will give you complete information. I hope the screen is visible.
Yes, sir.
Okay. So this information is what I got. Fine. Let me just close their COVID-19 information because it is mandatory these days. That's why they are representing. So we have closed it. So this kind of page will be appearing because I have searched specifically as gene. That's why this category came. If I will write Homo sapiens, then only Homo sapiens category will be coming. So the first category is the name of the gene, what you are talking about. Fine. And it's the description, what gene you are talking about, where it's located, and what are the names that are used to resemble this. Now you see here, I'm more interested in this part. Gene source, whether you're talking about genomic or mitochondrial or any plastid genome, you can go for specialized search in that case. If you're working on plants, if you're working on any kind of you want only coding, conserved coding sequences, yes, you can go directly to the CCDS database. If I open this, I just let me show you. We'll again come back to the CCDS database. See what it is giving me. It is giving me the same picture where the conserved domains have been showcased. Where the conserved domains have been showcased. So let's pick the Homo sapiens part, the tumor protein P53, Homo sapiens. First one was fruit fly. I am going for TP53. So you see, we had studied the P53 in Swiss prot and the gene name was TP53. And here also the gene name is TP53. So I'll open this. And you will end up directly on the P53 page. And the position is mentioned here, chromosome number 17, and we had already seen in that particular Swiss prot description where the chromosome was. So you have landed up on a descriptive page. You can see it's a descriptive page. Till now, it's not the entry GenBank page. Now you have to use this page to enter inside the GenBank if you are routing the way I have routed. Okay. So what it is doing? It is giving you a full report of this particular Homo sapiens 53. So what it is doing? See, it is giving you a gene ID. It is also showing you that recently this page has been updated with the information. So what you are getting completely updated information in these databases. So what's the symbol? Will give you a symbol, they will give you the official name. These are the black ones are the annotations, the right side are the descriptions. Whether this sequence is reviewed, yes, it is reviewed. Whether this gene that you are searching is actually a protein coding gene or it's an RNA coding gene, it's a pseudo gene, what it is. Then you can see here, this is a protein coding gene. Then they will give you the complete lineage also and a brief executive summary of this particular gene, what it codes for, what are the functions of P53 that it involves in apoptosis, senescence, repair, and so on, what we have studied in SwissProt database, right? Then what else? See here, expression. It is you where it is expressed inside our body. It is ubiquitously expressed in spleen, in lymph nodes, and other 25 tissues. You want to read more, you can read more. Orthologs. Now, orthologs are the proteins that perform same function in different species. So if you want to go directly to the mouse 53, you can go from here. There's no need to surf ahead. Now, when we are talking, this is a generalized summary, right? Now, when we are talking about the genomic context, when we are talking about the genomic context, let's see. Location is there. We had seen this location in while studying SwissProt also, right? 17th chromosome DR. Then how many exons are there? There are 12 exons in this particular gene. There are 12 exons. And the complete genome structure is represented in this particular part. That chromosome number 17 image is there. Now, let's not go to the image if it's complicated for you. Let us directly dive to GenBank. Now, in this section, you see here the position and it is written GenBank. GenBank. So this is the basic report that you can use. Let this these are all the cumulative informations that is there. You can directly go and see the GenBank part. And once you click the GenBank, you will be landing up on the principal page. You will be landing up on the principal page. See, this is the page. So we'll start this description of the page in just a moment. When we had typed the GenBank, we had landed up on a page which was saying for GenBank overview. So what is GenBank? You can read it completely. Already you had a very good understanding. Now, if you remember, while using NCBI, I selected gene and then I typed P53 here. Right now, I am selecting nucleotide and I am writing P53. The list pattern has changed completely, right? Now you see the list is completely different. The first one is P53 of mouse. It's not the gene. Please see the description. It is mRNA. That is the difference. In gene, you will always get DNA sequence. In nucleotide, you will get DNA and mRNA sequences, both. So what's your job? You want to retrieve the DNA sequence. So you have to identify where is your Homo sapiens gene expression. Homo sapiens gene expression, and then becomes a bit tedious because it contains 12 different exons, and every exon's partial CDS will be available here. Every exon coding sequence will be available here. So that is the reason why you should always navigate via gene. You should always navigate via gene, and you will land up on the page what we have used. You will always land up on a page that we have shown you. So let's go to a GenBank scenario. Left side annotation, right side description. First one is locus and then number, accession number, the length of the gene, that is 19,000 base pairs, 19,070 base pairs. It's a linear form, and the information was updated on 16th May of this year. Updated on, fine. By definition, what kind of gene is this? Then you will be getting multiple annotations, keywords are there. Then what is the organism source, whether it is homo sapien or mouse. Then the literature part will be there. What are the publications that were used to identify this gene sequence, how this sequence was identified. Then you will be, you can take the reference here from PubMed and can read how this sequence was identified. Then we'll shift here directly. Now the name is huge. Why? So many people have contributed for this particular publication. Why? Because the sequence was identified in Human Genome Project, and that was a mega project. People were working across the globe. Now, see, consortium where it was identified. It was identified in International Human Genome Sequencing Consortium, which is nothing but the Human Genome Project, and that is the link to the publication of Human Genome Project. So let us go to the feature table. That is the most relevant part of any gene. Feature table will define you about the gene. It is giving you the source, that the length is 1907, the complete description, whether it is genomic DNA or it's an mRNA. And then it will be talking about the gene. The gene name is T53. What are the synonyms that are there in literature, or what is the effector name? What is given? It is tumor protein P53. After that, main things start coming. After that, main things start. Now you see, as I am scrolling, I am seeing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 mRNA annotations written here. How a gene can have 15 different mRNA? See, or because of the splicing part. We had studied this splicing in Swiss prot also. The isoforms that were there, and we had a very clear-cut understanding how many isoforms were there for 53. And those isoforms are generated because of transcript variant, arising because of RNA splicing. And that is the reason why you are getting different mRNA sequence, and every isoform or every transcript variant sequence will be available here. Every transcript variant sequence will be available here. So starting, please focus on this particular part. Let me zoom this for you if it's possible. Yeah. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 10, 11. So we have 1, we have 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. What are these regions? These regions are the regions which are joining together to form a mRNA. Let me use this particular scenario here. What it is trying to say? It is trying to say, it's a very clear-cut scenario. Please try to understand. I have a gene here. This is 5 prime end. This is 3 prime end. Fine. This is my exon. This is my exon. This is my exon. This is my exon. Suppose. So the mRNA. This is my starting codon. Suppose this is my starting. mRNA will be formed for complete portion. Fine. mRNA will be formed for complete portion. This is immature mRNA, or we should call it as pre-mRNA. It will be undergoing splicing. And will be resulting into mature mRNA. What is mature mRNA? It will be having only these regions which are pertaining to the exon part, and the intronic region will be removed or spliced off. So effectively, you have a mRNA which contains all the 4 exons continuously without any gap. This is mature mRNA, right? So that is the thing that is being showcased there. From 1 to 100 amino acid, from 130 to 200 amino acid, the second exon. So the listing or the nomenclature that is given there, so the listing and the nomenclature that is given there in this particular section is saying the first exon is from 1 to 114. The next exon is coming after 10,870 first basically. So that is the thing that is being showcased here, and these are the transcript variants. So if you see the second sequence, if you see the second sequence, you see again you will be seeing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 11 exonic regions that are getting combined. Then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Again 11 sequences that combined. Now if you see the variation, if you want to see the variation, see 1114 is same. Again, its second one is same, third one is same, fourth one is same, fifth one is same. Then where is the difference? You will come to know once you compare the complete part. Once you compare the complete part. See here. Once you compare the complete part. And if the difference is there, they will showcase the difference in terms of the gene ID. In terms of the gene ID, that yes, the gene ID is 7157 and the variation that you are seeing may be represented in this manner. May be represented in this particular manner. So that is how, if somebody asks you that please pick up the exonic sequence, you can pick the exonic sequence from this particular section. You can pick the exonic sequence from this particular section. If somebody asks you how many transcript variants are there, you can count the mRNA and you can give the transcript variant. You can count the mRNA, the transcript variant, and if you cannot predict from that, you can easily see how many transcript variants have been given. It's the fourth one that we are talking about. Similarly, in writing, it will be given. Now see this transcript variant 3. It contains 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are complete 12 exons in this particular one. So that is how we decipher that what are the isoforms that are present at genetic level and variants that are present at genetic level, which will later be converted into isoforms. Now, once you are through with mRNA, then a section comes as CDS, coding sequence. Coding sequence, which is exactly your coding region out of all the mRNA what you have studied, which actually codes for protein. So the first coding region starts from 10897 base pair. That means the first amino acid will be coding starting from here. What was there then? Let me see this. Yeah, what was there from 1 to 114? Those were UTR regions, untranslated regions that are present in mRNA, untranslated regions that are present upstream at the 5' end. These are called as 5' UTR. These are called as 3' UTR, and that is the reason why you are seeing that at starting is at 10897 and termination is at 1782. After 1782 till 19070 is just the 3' UTR region. So that is a form that you should remember, and they will also tell you that among all the transcript variant, this coding region, which is actually the principal coding sequence, is derived from which isoform? This is isoform A, which is coded by transcript variant 1. And the sequence you observe, the first sequence you observed in UniProt of P53 was derived from transcript variant 1. And the sequence is also being showcased here. This is what? This is protein sequence, the same sequence which you saw in UniProt, the same sequence which you saw in UniProt. And now, another important part. See this region. These are giving you the links. If you want to see the gene ID, click on this, the ID will open. If you want to go to the conserved database, the link will open and you see the image will come. CCDS report. This is called as consensus CDS protein set. We'll come to this in a while, but again, you can find the report presented beautifully in this manner. You see what they are showcasing. The complete nucleotide sequence is there and corresponding protein sequence is there. The regions which are highlighted in blue, what they are indicating. they are indicating that these are the alternating exons. These are the alternating exons that are present in this particular protein. And the sequence, portions that are highlighted in red, the portions that are highlighted in red, these are indicating the amino acids that are at the splicing junction. You know, understand the RNA splicing. At the splicing junction, the amino acids that are present, there are those amino acids are encoded in red color in this particular section. So you see, you can get the complete coding sequence of that particular section which you want to predict, the CCDS sequence. So what is the thing? Out of out of the size of this particular out of the size of this particular complete gene, the size was 19,070 base pair. Out of 19,070 base pair, only 1182 nucleotides are responsible for coding 393 amino acid. Rest is what? Rest is introns, rest is UTR regions, rest is promoter function. Fine. So this is how we predict the exact coding sequence, and this coding sequence is accumulation of let me showcase that it's accumulation of these digits, It's accumulation of these digits, and you can easily go to the CCDS site and pick up the sequence directly from there for coding region. Then, likewise, for all the mRNAs transcripts, you will be seeing the coding sequences which are actually being converted into protein form, and corresponding CCDS report will be generated, and you can easily read where it is, what length of the amino acid is there, what is the exonic part, what is the exonic reach, what is the splicing amino acid junction present at the junction. So moving ahead, let's go to the segment and see. This is origin, and this is giving you the complete sequence of 19,000 bases in the form of ATGC, and this is called as flat file format. Fine. So this is what 10, 11 to 20, 21 to 30, and so on and so on. So when I'm saying that you have to retrieve the sequence, this is the sequence you have to retrieve, but always you have to retrieve in FASTA format. You have to retrieve in FASTA format, and you click on FASTA, and here you go. Starting from greater than sign and the accession number, you will get the complete FASTA format sequence. You can take this sequence and you can go ahead. Right now, majority of the time students says, I ask the students, please pick uh, first 72 amino acids. First 72 amino acids. So they'll start counting 1, 2, 3, 4. No, that's not the way you should do. No. See here, window is there. Selected region is given to you. Selected region is given to you. You just add 72 here. You just add 72 here. You add 21 to 72. It's 93, right? It's 93. Control A, Control C. Type here. 93. Update view. You got the sequence. So whatever the region I want to pick, I can pick in this window. You need not to count it. That's the thing that's been provided to you. Whole sequence you want to see updated, you will get the complete sequence of the chromosome. You will get the complete sequence of the chromosome. You want to go for selected region, pick it up, update the view. You will be getting the complete updated region. Fine. So that is the thing that you should remember, that we can pick FASTA format from this section directly. Now, coming to this part, what we have seen gene, RNA, and CDS. If you want all the features that are provided, you can click all and update the view. So they will be giving you the complete part. They will be giving you the complete information what they've hidden. If there is any, see, miscellaneous features has now started coming. What are these miscellaneous features? They are the links that are there for different literature, for UniProt and SwissProt, links will be there. So all those miscellaneous features will be starting coming if you are interested in those features. But most of the time, we are not interested in miscellaneous features. Why? Because we are surfing the genomic database and we are interested in DNA, RNA. coding sequence and its prominent sequence. And this is how we retrieve the complete sequence using GenBank. Using GenBank. Okay. So then, furthermore, what is there? Furthermore, what is there? On the right hand side, there are multiple links that are there. There are multiple links that are there that you can explore via NCBI portal. You can pick primers. We'll be telling you how to design the primers. Yes, there is an exercise for you in this session that you are running PCR. You are not aware of the primer. Then how will you design the primer? That will be showing you in the coming lectures. So the complete link is explorable. You can explore it. Now, that is all for GenBank. That is all for GenBank. So let us go back to the our PPT and let us try to understand what we want to showcase here using this sequence. Now, if you are using this sequence, what you can do? The first thing you can do is alignment. Your job is to identify in lab. You are working in lab. You are working in lab. Suppose you are working on microbial aspects. You went to a niche, you isolated the microbial colonies. There are plentiful of microbial colonies. You were working on, let's say, in an industry of fermentation industry, and you have isolated a novel strain, a microbial cell, which is actually giving you a very good production of any antibiotic, let's say streptomycin. Let's say a good yield of streptomycin you're getting. Now you want to see whether this isolated microbe is new or it's already known, and if it's known, to which species it belongs, what you will do? You will take out the DNA. Fine. You will go for PCR using specific primers. Now, if the genome is too large, you will not be amplifying the complete part. You will be using the primers which are specific against the specific marker. So suppose you have taken the 16S rRNA sequence. You are talking about 16S rRNA sequence. Or we have taken the ITS region if it's a fungus one, ITS1 primers. So I have amplified a small stretch of amplicon. So what I'm getting, I get substantial amount of DNA of the selected region of an unknown microbial source. I will sequence this region. Now I have a complete sequence in the form of AT, GC, GC, and so on, belonging to this region only, not the complete part. What I'll do, I will take this sequence and go for database searching via alignment. I will use the tools which will match this particular sequence one by one or simultaneously to all the microbial genome sequences that are present in the database and give me the result. If I get a suppose 90 to 95% similarity, that means the species is already known, it's belong to this. Suppose I get only let's say a 60% similarity, that means it's a new species that has diverged from this particular ancestor. So all that information, all your claim on that particular species will be coming after analyzing the bioinformatics tool and after processing with the alignment part. Whether it's DNA, whether we are talking about any protein, whether you want to study any evolutionary pattern, whether you want to identify the site where you want to go for site-directed mutagenesis, alignment is very, very necessary. So what we do in alignment, just to give you, and I'll not take much of your time, just to give you this, what we do in alignment. This is my query sequence. Which I have taken. This is my database which contains which contains multiple sequences, which contains, let's say, for now, let's talk about 4 sequences. I want to see my query is resembling to which sequence, whether it is similar to A, whether it is similar to B or C or D. So what I'll do, I'll match the sequence one by one. Fine. I'll match the sequence one by one. And after matching, I might see that Q is actually similar to C. And that is what we are doing. This matching is actually called as alignment. And if matching you are doing manually for 1, 2, 3, 4, okay, you might do. But what if this number goes to 10,000, or it goes to lakh, or it goes to 10 lakh? It's impossible for you to do manually, right? So that's why there are specialized softwares, specialized tools which will be taking out and doing this job for you one by one. Also, simultaneously, also based on that, we have two types of alignment process: global and local, pairwise and multiple. And that complete detail we'll be studying in the coming lectures, where we'll be first studying the alignment, then we'll be talking about the blast, which is used to perform the alignment, and FASTA, that is used to perform the alignment. These are the two basic softwares that we use. Then we'll go to multiple alignment and we'll see cluster first of all, so that you have a very decent exposure to the basic things. And then we'll take the information and we'll go to advanced part of Mega, Mr. Base, and Fire. We can use Fire also. Fine. And then we'll be going directly to the advanced part of PCR based designing, docking studies, and so on. So till now, we are an inch closer in having a very good understanding of alignment part. So next lecture, that is on Monday, that is the next lecture, we'll be talking about the alignment. And I hope till now what we have done is pretty much clear to you. So thank you so much for your patience listening, and
Thank you, sir.
very
Thank you, sir.
good.